So again, thank you everyone for joining us today. Just gonna pull up my slides real quick. So as I said, this is the Open for Neuroscience tutorials, tools for modeling. Again, please keep those introductions rolling in. Here's a brief overview of what we're going to be covering today. At any point, please put those questions in the Q&A, and we will save most of them to the end unless you need immediate clarification on something. And we'll also post the Q&A on our community forum after the tutorial ends. So we're going to start with an introduction to modeling tools from the Allen Institute with Anton, and then we'll have a live demonstration of the Brain Modeling Toolkit and SONATA data format with Kale. And again, we'll have time for questions at the end. So here's a little bit about orientation. Please put those questions in Q&A. Unless you're on YouTube, just ask them in the chat and use that chat to talk to each other. And make sure that you're sending your chat messages to all panelists and attendees. We have live captions available on Zoom. Go ahead and hit that CC button and then captions, or on YouTube for viewers of the recording. Our presenters today are Anton Arkhipov, an associate investigator at the Allen Institute, and Kale Dye, a software engineer at the Allen Institute. Both of them were involved in the development of the resources they're going to be sharing with you today. So the Allen Institute is a biological sciences nonprofit located in Seattle, Washington. Our focus research areas are neuroscience, cell biology, and immunology. And we support cutting-edge research across the world with the Paul G. Allen Frontiers Group. Across our institutes, our scientific process is focused on tackling complex, broad, and hard problems in the fundamental sciences. And our approach is guided by 3 principles: big team and open science, which I will return to in a moment. And our work generates data, knowledge, and analysis tools like the modeling tools we're going to talk about today, all of which we share publicly. And to date, we have collected about 14 petabytes of data, which is about 2.8 million DVDs worth. So those 3 core principles are team, big, and open science, the most relevant of which today is open science, which is to say that we share everything that we create in our research, all of the data analysis tools, all of the data that we generate publicly for you to use in your work. So again, please check out some of the other webinars in this series to learn about more of our open resources, and come back in March 8th through 10th for the Open for Neuroscience Symposium to hear some talks about more resources, how they're being used in research, and some more tutorials. And we will get the link to register for that in the chat. So I am now going to hand it over to Anton. Again, please use that Q&A button in Zoom to ask questions so they don't get lost in the chat, but continue to use the chat to chat with each other. Make sure you're sending messages to all panelists and attendees. So Anton, take it away.
Great, thank you, Caitlin. Thank you. Welcome, everyone. It's great to have you all here today. It's my honor and privilege to speak to you. And today we'll talk about our tools for modeling, the Brain Modeling Toolkit and the SONATA data format. So let me start with a bit of a motivation for this work. And the motivation is basically that we want to establish and support a virtuous cycle of experiments and modeling. So especially it's important here at the Allen Institute, but it's a sort of an approach that is important for the whole of computational neuroscience, where what we want to do as researchers is to integrate different types of experimental data into models that will then make predictions. But especially for someone like me, a modeler here at the Allen Institute, this is really close to the heart, because we are collecting huge amounts of data, very high quality data. That's what the Allen Institute is famous for. So this data, in terms of brain structure and activity, they involve information about cells, different cell types, their properties, connectivity between these different cells and cell types, and also their functional properties in vivo obtained using electrophysiology or optical physiology. So it's a lot of different multimodal diverse data. And what we want to do, besides providing this data to the community and analyzing this data, we also want to model this data. And we want to maintain a high level of biological realism in these models. So this is what this arrow indicates. We are integrating this data into different types of models. I'm showing here the biophysically detailed and point neural network models, but different teams here at the Allen Institute are working also at other levels, at more abstract, more theoretical levels of resolution. But ultimately, it's the same idea. We want to integrate the data and run simulations and make predictions, right? So we make predictions from the simulations, and ultimately that should lead to new experiments for testing these predictions, and then you repeat this loop, and that's how big discoveries and a lot of progress will happen. So we've been working on this quest for several years now, and our latest achievement was published last year. It's a model, actually a series of multi-scale models of mouse V1, the primary visual area in the mouse cortex. So like everything else, all the data and tools that we produce at the Allen Institute, these models are freely available, can be found here on this portal. And let's talk about what went into these models. So first of all, it's the cells. Cells are the building blocks, neurons. So we have this database, the Allen Cell Types database, which contains morphological and electrophysiological intrinsic properties of neurons. We built models of individual neurons based on those properties. And then we use them as our building blocks for the model. We take those models and distribute them into the space which will be occupied by our network model in a realistic way. So the next one is connectivity. It's very, very important to connect cells in appropriate ways according to what we learned from the experiments or according to our assumptions where experimental data are not available. And for that, currently we have really amazing resources here on the same website, which I urge everyone to check out. So one resource is the synaptic physiology data set, which is a collection of multi-page slice recordings that provide connection probabilities and synaptic properties, weights, kinetics, short-term synaptic plasticity for many different cell classes in mouse V1. It's a really an amazing data set. Moreover, we now have the electron microscopy data set with ultrastructural dense reconstructions of the circuit in the mouse V1, which ultimately what we will release will be a full cubic millimeter of tissue in mouse V1 with about 100,000 neurons. We'll know who they are in terms of morphology. We will know who is connected to whom, so the precise connectome of that specific piece of the tissue. And that will be an amazing resource for modeling and analysis. And you can hear more about some of these resources actually in the next installments of this same series in about a month from now. So these resources are really great. We want to use them for modeling. We are using them for modeling. The models that we released thus far actually were based mostly on the literature as far as connectivity is concerned, on a very thorough survey of the literature. And the result of that survey, sort of the matrices that describe that connectivity, which is, our best guess at what this connectivity is at the moment, they are provided together with our models, also for free. Finally, there is the types of data of in vivo physiology, especially our neuropixels data set with recordings from 6 neuropixels probes simultaneously from multiple cortical and subcortical areas. Of course, for our model, the main sources that we want to look at are the recordings in V1 itself and the LGM, lateral genicular nucleus of the thalamus, that provides visual inputs into V1. And so a small part of this data were used to optimize the model, to make sure that the model behaves well, that the activity that exhibits is comparable to what we see in the experiment. And then the rest of this data was used to test the model. It's a test set, right? And we the important part of the model is that it can process arbitrary visual stimuli. So we can use any movie, any type of visual stimulus that was used in the experiment, and then compare apples to apples, the output of the model with what was recorded in the experiment in vivo. And we found that the model did pretty well on a number of those stimuli, maybe less well on some others. But this is an iterative process. We want to build better versions of these models with time. And ultimately, the result is here. So we have these models. This is an illustration showing where these models are on the mouse cortex. There are two versions. So there is a biophysically detailed version which uses compartmental morphologically detailed representations of neurons. There is a point neuron version. In each of these models, we have about 230,000 neurons, which is the majority of neurons in mouse V1, 17 different cell classes, and like I said, very realistic patterns of connectivity and so on. One-to-one mapping between these models. So there is a one-to-one mapping between neuron here and here, exactly the same connectome. And then, of course, for the biophysical model, one requires a lot of computing resources to run simulations with them, whereas for the point neuron, we can run it on a single CPU with about 3 minutes for one simulated second. So all of that is freely available here, and we really welcome everyone to use these models, modify them for your purposes, ask us questions, and give us feedback. Now, to make all that happen, we had to develop some of the software tools, and that's what we'll be talking about today. There is a good number of really impressive, powerful tools that exist in the field and have existed for a while. For example, software like Neuron or Nest. But we found that for the project of the complexity that we are talking about here, we had to develop some additional elaborations on those tools or some additional tools from scratch, especially for sharing our work with the community and running simulations across different levels of resolution. So one of the tools that resulted from this is what we call the Brain Modeling Toolkit. It's a free open source Python-based software package, which is available here on GitHub. And it's basically a tool for building models, for simulating models, and for doing some basic analysis. This picture illustrates the structure of BMTK, which has a builder module and then a few of the simulator modules for running simulations at different levels of resolution. So let's talk a little bit about the details. So in terms of the network building, we provide here in BMTK a Python APA for building and saving large-scale network models. So we can use that across different levels of resolution for biophysically detailed or point neuron models or population statistical models. The tool allows for custom attributes and functions. So we provide some basic functions for building models, for building the nodes and for connecting them. But importantly, it's very easily extendable if you want to add some of your own functionality. That's very easy to do. And Kale will show you later today how to do that. And then we save the output of this model building procedure into the SONATA format, so a standardized format, so that we can use it later and then other software can also use those files. And the basic parts of the workflow here is to establish the nodes, to decide what your populations are, provide the properties of the nodes, then determine the connection rules, and then instantiate the network and save the output into the files. And you'll hear more about this from Kay. All right, so now in terms of the simulators, the key functionality that BMTK permits is running simulations across different levels of resolution. And another important component of it is that it provides automatic support for parallel simulations. So tools like Neuron and Nest, for example, they have excellent parallelization capabilities. However, often to take advantage of them, you need to write your own code using their commands. And so what we've done here, we created an interface to those tools, which already takes care of that part where you don't need to write that parallel code, where that code exists for you and you don't need to understand it. You just use it as a black box, more or less. And of course, you can go into the inner workings of it, but you don't need to. So we can run simulations at different levels of resolution. One is the module that we call BioNet. It interfaces with Neuron to allow for simulations of multi-compartmental models, the networks of biophysically detailed neurons. The other one is what we call PointNet. It interfaces with Nest to simulate point neuron networks. The next one is PopNet. POP standing for population, and it interfaces with another Allen Institute tool called DeepD, which is a simulator to model dynamics of neuronal populations, right? So here we do not resolve individual neurons. Instead, we model whole populations of neurons as a single node, and they can be connected to each other. And then finally, there is the FilterNet simulator module, which operates with filters. So these are the mathematical entities which take in some kind of inputs. So for example, what we are typically working with are movies, spatiotemporal patterns of activity on the screen, right? A 3D, it can be a 3D array, so X by Y by time, which defines a movie that comes onto the retina of this animal's brain that we are simulating. So this module allows one to instantiate the filters that will process that kind of visual stimulus, and as an output provide to you firing rates or spike trains that you can then use, for example, as inputs to the simulation of V1 in our case. So that can be very useful for a variety of applications. And of course, the parameters of these filters need to be selected, in our case, at least based on the experimental data, and it all can be done. And there is a lot of diversity in applications of this tool, but it's there for you, and yeah, it can connect with all these other simulators. Okay, so let's now talk a little bit about the SONATA file format. So SONATA stands for Scalable Open Network Architecture Template, SONATA. So it's a free and open format and schema, which are all available here on GitHub. And originally, this started as a collaboration between us and the Blue Brain project in Switzerland. And then later on, additional colleagues got involved and contributed, representing the open source brain and NeuroML, NetPine, Pine, and NeuroData Without Borders. And so as a result, we have this format. We published a paper about it last year. So what is it? It's an efficient format for sharing models and simulations across software tools. So what it offers is the efficient ways of storing the data about your model and simulation results in binary files. Not only binary, but for the most data-intensive parts, we use binary files. It's mainly focused on heterogeneous and fully instantiated networks. And that's the key. You know, this is something that we have to deal with when we work with biologically realistic models. Typically, it's too expensive to regenerate your model every single time that you run a simulation. And also there is way too much detail to keep track of. And so for models like this, it really is necessary to have a data-driven format which saves everything, every single detail about your simulation. And that's what SONATA supports. And it's not a replacement for other formats, things like NeuroML, SWC, and others. It's not a replacement. In fact, SONATA is a hybrid format, and it uses some of the existing formats as a part of specification. For example, if you want to have neurons in your model with a morphology, you can do that using SWC files to define those morphologies, right? And SONATA will point to those files, and it will be a part of your model. So just to illustrate the basic components of it, here we are showing a picture where these are the major stages of the modeling and simulation workflow. So we have a model network, which consists of some nodes that are in different populations. They are connected by edges. There can be external virtual nodes, which we are not simulating explicitly, but which may provide inputs into our model, like spike trains. Then we run a simulation. We need to specify how long the simulation we want to be and some other conditions. And ultimately, there is an output, which is typically spikes and time series. So SONATA covers all of these parts. And in the SONATA representation, this is what we have. So there is the part of SONATA that takes care of nodes. It saves the information about nodes in CSV and HD5 files. There is a part of SONATA that deals with edges, also saving information into CSV and HD5 files. And those nodes and edges, those files can point to other parameters such as morphologies and biophysical properties of the cells that may be saved in some other formats. The division between CSV and HDF5 is basically, the idea here is that some properties may be repeated among the cells and they may be more like a cell type level property, and other properties are individual for each cells. And so those repeated properties, it might be easier, more convenient to store in a text-based format in small files, whereas the individualized properties can be stored in binary files because there are a lot of them, a lot of cells, a lot of connections. And there is a flexibility. It's up to the user which goes where. Then the part about simulation, we have the parameters stored in the JSON file that determine things like the conditions of your simulation, like the temperature and then how long it should run and so on. And then finally, the output is saved into HDF5 files. The output can be spikes or time series associated with the neurons or compartments of neurons, or it can be time series that are associated with some other things. Like, for example, we may want to store extracellular potential, electric potential associated with electrodes rather than with individual neurons. And SONATA can do that as well. All right, so SONATA already has a pretty vibrant ecosystem, which is illustrated here. So due to our collaborations, we have now several tools that can produce SONATA network files, some libraries that can read those files, and then some tools that run simulations and even something that can do visualization and produce SONATA simulation output or reader types of output. So a lot of tools, and as far as we know, there is more interest here, and there will probably be more tools supporting it in the future. This is also compatible with NWB. So NWB is a powerful format for representing experimental neurophysiological data, such as spikes or time series from optical physiology experiments, for example, or neurophysiology, electrophysiology experiments. So we work together with that team to create converters between SONATA and NWB. And so now we can go between these two formats in terms of neurophysiological data. All right, and before I end, I just want to mention something about the future, maybe to keep you all excited about it. So this is another tool that we are starting to work on. We call it Visual Neural Dynamics, or VND. And this is a collaboration with this group at the University of Illinois, very, very talented people with an established, very powerful tool called VMT, Visual Molecular Dynamics. So the point here is that when we work with very complex, biologically realistic models, it's important sometimes to keep track of what's happening in this model and simulation using our vision. As humans, we are very good with our vision, and so visualizing things in 3D can be very useful. There are tools out there that can do this for the types of systems that we are working with, but there are typically a lot of limitations, like especially if you want to visualize a really large system of neurons, that can be difficult. And so we are leveraging this existing platform with excellent visualization capability and with very efficient way of doing visualization of very large systems. And what we're working on is that this new tool will be able to read SONATA files. It will perform efficient 3D visualization on regular workstations and on specialized hardware. So from simple computers to high-end workstations for rendering. And we will be able to produce movies of neural activity, 3D maps, rendering of devices, such as this NeuroPixels device going through our model of mouse V1, or such as those movies of neural activity. So stay tuned, and we hope to share with you more in the future. All right, so to conclude, we have these resources. We are sharing them with the community. We are very excited about people using them. There is the Brain Modeling Toolkit on GitHub here, SONATA on GitHub here, and our models are freely available here on our brain-map.org website. So please come use them, try out our resources, let us know what you think, tell us about the features that you want, and we'll be happy to engage. So thank you for your attention. With that, I'll thank our founder, Paul Allen, for his vision and getting us all into this very special place, the Allen Institute, doing very important work. And for all the colleagues at the Allen Institute for doing all the amazing work. And now I'll pass it on to Kale to do a hands-on tutorial. Thank you.
Kale, you're muted.
Hello, sorry about that. I assume you can see my notebook. So thank you, Anton. As he said, my name is Kale, and I am the primary software developer for the Brain Modeling Toolkit, or BMTK. Today, I'm going to go over a high-level overview of some of the core features of BMTK and the SONATA data formats. There's a lot to cover, so please excuse me if I go a bit too fast or skip over parts of this tutorial. If you want to follow along at home, this notebook and all the associated files can be found at this link, which is our BMTK GitHub repo under docs/tutorials, where you can also find other tutorials, documentation, and examples. So the first step is going to be to install the BMTK Python library. You can download the latest version and install it from GitHub. We also have pip and Anaconda packages to install it. And for parts of this tutorial, we're also going to need to install Yale's Neuron Simulator libraries. You can download Neuron installers from their homepage or use pip or Anaconda to install it. However, we also do have Docker containers with BMTK and all the prerequisites already installed, including Jupyter Notebook with this particular notebook. So if you have Docker installed on your machine, you can do a Docker run command, the Allen Institute /BMTK container, and you can even start up a Jupyter Notebook instance, which includes this tutorial for you to follow along and play with at home. So the goal of this tutorial is going to be to build a small toy network of the layer 4 of the mouse primary visual cortex and simulate it across a variety of different conditions. The actual layer 4 model, which can be downloaded from the Allen Institute data portal, contains 45,000 cells. And like Anton said, will pretty much require access to a multi-node cluster to build and simulate. The model that we're going to build here will only contain 300 cells and generally should be small enough to simulate on any modern computer or laptop in a reasonable amount of time. So before we can actually run a simulation, we must first create the SONATA network files, which, as Anton said, we can do using the network builder component of the BMTK. So first step is in a Python script or using Jupyter notebooks. We can start by importing bmtk.builder.networkbuilder. Then what we're going to do is instantiate a new model instance, which we'll call V1 here, or primary visual cortical area 1. And then usually when we're doing any kind of modeling, first thing we always have to do is sort of figure out what cell types, which are also called node types in SONATA parlance, we want to use. And when we sort of determine this basic foundational structure of our network model, we can go ahead and start using the network builder add nodes method to actually start adding cells. So here, as we can see, what we're doing is calling v1.addNodes to add 80 SC and N1A type excitatory cells to our v1 network. Most of these parameters, like model type and model template, are actually going to be reserved SONATA keywords, and they're used to indicate how to actually build and place cells during a simulation. And because they're SONATA standard, they can even be used by other tools that are SONATA compliant. However, some things are not reserved keywords, but will be germane to actually describing our particular model. So for instance, here we see we're assigning each cell a different tuning angle, and which is a property found in excitatory cells of the primary visual cortex, but wouldn't necessarily be applicable to other cell types or even other areas of brains. Instead, they might have other properties or attributes that they want to use instead. So this is highly customizable. So all we have to do is call add nodes. And here we'll call v1.addNodes a few more times, where I'll add, calling it twice to add 2 more groups of excitatory type cells. And then finally call add nodes once again, last time, to add a group of inhibitory type cells. And in total, these 4 method calls will give us will give us 300 individual cells across 4 different cell types, with 80% excitatory and 20% inhibitory. So now that we actually have our cells, or nodes as we call them in SONATA, we can go ahead and start creating our synapses or SONATA edges using the add edges method. When we call v1.addEdges, the first thing we need to do is pass in either a list or a dictionary of conditions to determine what cells we want to use for our presynaptic source cells, and what subset of cells we want to use for our postsynaptic targets. Here, so we can see we have source and target properties, and here what we're doing is we're using the EI property, which is a custom user-defined property that we are using to assign cells as either excitatory or inhibitory, to create type of connections that will connect all the excitatory cells to the inhibitory cells. We also have properties like syn wait or delay, which are once again SONATA attributes, SONATA reserved keywords. But just like add nodes, we can create our own custom attributes and properties. So when the network will actually get built, what it's going to do is create a directed multigraph. And what that means is between any source and any target, there can be 0, 1, or more connections between them. And so to specify how many there are, we're going to use a special connection rule property here. And here we're going to use a connection rule property that just has a scalar value of 1. And what this means is that between every possible source and target cell, there is exactly 1 synapse from each source to each target. We'll do similar, call a function to create edges between excitatory to inhibitory. But with this connection rule, sometimes we, besides just passing in a scalar value, we can also do is pass in a matrix or an array of the appropriate length. Or even better, what we can often do is create our own custom functions to intelligently determine the number of edges between each source and each target, which Anton sort of briefly mentioned before. So here we're going to add create connections for inhibitory to inhibitory type connections. And to do so, we're going to create this own custom function called ignore autopasses. And mainly this is to prevent a cell from synapsing with itself. So what this function does is it checks to see if a cell, the source and target cell has the same ID, then it returns 0. Otherwise, we just return a random number of connections between them. And then to use this rule, all we have to do is pass in the function name to the connection rule, as we do here. And when it's the connectivity matrix starts getting built, it will see this rule and sort of use it to build the connectivity matrix. Similarly, for excitatory edges, we can use a bit more complex rule called tuning angle, where the number of synapses is calculated based on the difference between the tuning angle properties of both cells. So what this function does is this connection function does is it looks at for every source and every target cell, it's going to take this tuning angle property, which is just a value between 0 and 360 degrees. Then we're going to sort of calculate the distance in degrees between the source and the target, or the tuning angle distance. And then based on the distance, we will randomly return a number of connections, so that the closer that the cells are in tuning angles, the more likely they are to connect. And this is somewhat of a rule that's been somewhat observed in actual models of Cortex. And then once we've created this custom function rule, all we have to do is pass it into the connection rule, and then we're good to go. But and finally, so we've determined what rules we want to use for building nodes or cells, and what rules we want to use for creating edges or synapses. And then finally, what we can do is we can call the build methods and the save method. And what this particularly is going to do is it's going to create the SONATA files and save them into the network directory. So it's already built for you, but if you can run it again from your home computer. And what it's going to do, it will actually sort of determine the names of the files for you. But we can see after we call build, we're going to have these V1 nodes and V1 edges SONATA files. And so so we can see these are actually our files that determine our V1 model. But before we move on to actually simulating the V1, we're going to create another network, which we'll call the LGN. So the LGN is actually a part of the thalamus that provides much of the input into the primary visual cortex from the retina. Our LGN model is primarily going to be used to drive the V1 during the simulations. So what BMTK and SONATA actually allows us to do is to build parts of a network independently and connect them together during this simulation process. So this section of code, which just creates a network of LGN cells, 100 LGN cells, and connects them directly onto the V1 cells. When we build it, it's going to create these LGN and LGN to V1 edges. What we can do is, by sort of building them independently and connecting them together, that means if later in the future we wanted to say remove the LGN inputs or replace it with something else, or even had higher cortical areas, we can do so pretty dynamically without having to rebuild the V1 model again every time. And one last thing to note about this is for the LGN cells, we're using model type virtual, whereas for the V1, we were all using model type biophysical. Virtual model types are a special designation in SONATA for cells that act as placeholders for spike trains or preset voltages. They cannot be recorded to or synapsed onto, but what they can do is actually synapse onto biophysical cells to sort of activate them. So now that our network model is built, we can go ahead and set up and start running our simulation. As Anton mentioned, BMTK includes a number of tools for simulating across different levels of resolution. For our network, we want to use multi-compartment biophysically detailed cell models. So we'll use BioNet simulator component. If we were starting from scratch, we would need to do some setup, including downloading external files and creating the configurations. There are scripts that can sort of help you do this and sort of build a basic foundation for you to start simulation. In this case, we've already sort of downloaded everything you needed and set it up here. Pretty much everything that's going to be required to actually instantiate and run a simulation is going to be saved in a SONATA config. What these are just simple JSON files that you can pretty much open up in your whatever your favorite text editor. Here we're going to call it config.bionet.json, and we can see that it's sort of divided into sections, different sections to help you instantiate and run a simulation. So here we see the first look at the network sections. This is where we actually specify the location of the files we had the LGN and the V1 model files we previously created. And like I said, if we wanted to remove parts or add in other parts of areas of the brain, other models, we can do so here. The components section of the SONATA file is where we can specify location of third-party files like SWC morphologies or NeuroMel or JSON cell dynamics. Then we have the run sections where we do the runtime parameters and conditions. Most importantly, we have T-start or the start time, the stop time, and the time step parameters. So what we're doing here is we're specifying we run a 2000 millisecond. By default, SONATA always uses millisecond values. Just so this is going to be a 2000 millisecond or just a 2-second simulation. For the outputs and reports sections, these are actually where we specify what we're going to record and where we're going to record it to. By default, Bionet will always try to save the biophysically detailed V1 cell model spikes. And here we're going to direct it to actually, when it saves it, it's going to save it under output directory under a file called spikes.h5. And then for reports, we can also do other things besides spikes, like calcium, or here we're creating a new report called membrane potential to actually record the membrane or membrane voltage or V variable of all the SCNA1 A cells. And then finally, we have the input section where we tell Bionet what stimulus we want to use to drive our model. You can include other stuff like extracellular stimulus or current clamps. And as I said before, here we're going to drive our model using the V1 cells, but they're virtual, which means they don't do anything by themselves. So what we do is we're instructing it to actually pass in a spike trains file, or here we're going to call it LGN spikes.poson.h5, which just contains a bunch of preset spike trains for each of the LGN cells. For that spike trains file, there's multiple ways to generate it. Here's an example of how to actually generate one of these spike train files, the one we're going to use. What these 2 lines of code is actually going to do is create a, for each of the 100 LGN cells, it's going to create a bunch of spike trains firing at 8 Hertz, generated using a Poisson distribution. And so finally, now that we have our entire configuration set up, we can go ahead and actually run Bionet or run the V1 model. We can do it through the command line using the run Bionet script. It also has support for MPI or parallelization. So if it's a very large network, we'd often need to actually distribute it across multiple cores to run a single simulation. But in this case, this is a fairly small network, so we can just run it using these few lines of code in Jupyter Notebook or a Python script. And what these lines of code do, we'll actually instantiate it, run it, and save the outputs into the output folder. So as I said, in the config, we were saving the spikes.h5 and the membrane potential.h5. And if you run it again, you can get new versions of it. So here are the actual results. Since they're SONATA, you can pretty much plot them in any SONATA compliant tool. Here we're plotting the rasters of all 300 V1 cells across the 2-second simulation. We can see how each individual cell, as well as each individual cell type, reacted to our Poisson input. And also, as I mentioned before, we're also recording the membrane potential of all the SCN1A cells. Here, the gray are actually all of the 80 membrane potential traces, while the blue is going to be just the average of all the membrane traces. Now, if we wanted to simulate again using different stimulus or reports, all we really need to do is change the configuration JSON file and rerun the script. So for example, a very useful tool in neuroscience is the ability to perturb a subset of neurons and record how the other neurons will react. And we can simulate this behavior by adding a current clamp to the config. So here what we're going to do is, if we open up the BioNet config, we can add in a new section into the inputs, which we'll call inhibitory or INH perturbations. And what these few lines in the JSON file are going to do is instruct BioNet to actually add in a large depressing current clamp starting at 500 milliseconds just into the NR5A1 cells. And then once we've modified our config file, we just have to rerun BioNet as before. And like I said, as before, it's going to save the output into the outputs folder, and then we can just plot the results. So here are the results from running the V1 model with by inhibiting the NR5A1 cells. As we can see, not only after 500 milliseconds were we able to actually silence the NR5A1 cells, but actually doing so had a very noticeable effect on the firing rate dynamics of the other cell types. And also just also mention, if you wanted to, you can also add in more than one current clamp input. You can add in like multiple current clamps, either in serial or even have them go off at the same time. Another easy thing to do by modifying the config is to add a extracellular recording. So if we went to the report section, we can add a report like this called ECP. And what this is going to do is it's going to actually insert a multi-channel extracellular linear electrode into the middle of our circuit. Something that would sort of look like this, although this is the more advanced model. But then we run it as before. And what the ECP report is actually going to do is create a new type of output, extracellular field report, where we can plot the extracellular traces for all 10 channels in our electrode file, from our electrode file. There are some features of BioNet, however, that may require a bit of programming. So if you may recall, when we were building our edges, we were using a constant value for the synaptic weight parameters, or syn weight parameters. What that meant was for every synapse, for the synapses for every edge type, it will be the same strength. There are a couple of ways to make a network where all the synaptic strengths are different. We can do so through the network building process, or alternatively, we can dynamically adjust synaptic weights during the simulation process. So here what we're going to do is create a custom function called setSynWeight. And what it's going to do is it's going to take in for every edge, for every edge type with every source and every possible target. It's going to look at the original synaptic weight or syn weight value. It's going to get the coordinates of the source and target cell, calculate the distance, and it will adjust the synaptic weight and return a value that's adjusted based on the Euclidean distance between the source and target cell. Then to use this function, all we need to do is call BioNet.addWeight function to actually register this. And then when BioNet is actually instantiating our network, it will see this method or see this function, use it to actually adjust the synaptic weights and run everything as before. And then we can see we get different results by adjusting the synaptic weights. Here it actually sort of seemed to, we strengthen it. So we get a lot more firing in as we did before. And there's a lot of other features that BioNet that we just don't have time to go over, but I would recommend take a look at our documentation. So far, our simulations of the V1 have been driven using spike trains generating using a Poisson process. This sort of stimulus based on simple mathematical functions can be beneficial for discovering fundamental properties of our network. However, if we want to compare our model to actually recorded data, like the Allen Brain Observatory data, it'll be useful to create more realistic input. And this is where, as Anton said, the FilterNet simulator comes in. What FilterNet does is allow us to play a movie or other visual stimulus onto a field of neurons. It will then convert the receptive field into spike trains based on the spatial and temporal dynamics of the input. And then we can use the results to get, we get from FilterNet to drive our V1 simulations above. So running FilterNet is very similar to BioNet. Most everything we're going to need is going to be saved in a SONATA config file. Here we're going to call it config.filternet.json. It has the same structure as the BioNet config, only it's much simpler. So if we notice in the nodes section, we can specify, we're only specifying to run it against the LGN cells. FilterNet can't actually do any biophysical detailed simulation. We'll still need to use BioNet for that. For the input section is where we specify, as I said, our visual inputs. We could create our own movies, but FilterNet also comes with a few preset stimuli. Here we're doing a full field flash. So what we're going to do is between 500 and 1500 milliseconds, we're going to flash or pretty much darken the screen for 1 full second. And then finally, we'll make a few changes to the output section just so we aren't overwriting our previous results. And then once we modify our config, as before, we can run the simulation, FilterNet simulation, either through the command line or we can just run it through the notebook. And then here we see the results we get from FilterNet. It will automatically save the results. So when we were building our FilterNet model, we created it using a subpopulation of simple LGN on cells and simple LGN off cells. And looking at this output of FilterNet, we can see how this flash, this dark flash, the effect it had on both the on population and the off population. And then finally, to sort of come around full circle, what we can do is we can take this results, which we saved under a file called LGN spikes.flash.h5. We can make a few modifications to the BioNet config. So replace the Poisson inputs with the flash inputs, and then run the V1 model in BioNet again, and we can see the results we get by running our V1 model in with this new input. And I am definitely running out of time. As I said before, we could also do more advanced stimulus, like playing our own custom movies. But since I'm running out of time, I can't really get into it anymore. But I would suggest you look at, we have a lot of other documentations, some other tutorials with more and documentations, especially at our alleninstitute.github.io/BMTK pages. And as I said before, I apologize for going a little too fast, but you can follow along at home, which I encourage people to do. So with that, I'm going to hand it back to Caitlin. And thank you very much.
All right, thank you. We do have time for a few questions, and we'll make sure to post all of the questions and answers in our community forum afterwards and get the answers to any questions that are still lingering at the end of our time here today. So you'll be able to find that post in the description of the YouTube video, which we will get posted later today or tomorrow, and on our community forum at community.brain-map.org. So Anton has been answering a lot of these questions in the Q&A panel in Zoom already. So those of you who are viewing on Zoom, you can start reading some of those answers there. And again, we'll post those after the fact. So let's go ahead and start with the question from Florian. As a Nest user with my own custom build synapses, studying various new forms of plasticity that are not part of mainline Nest distributions, I'm curious to see how I can look under the hood of generated Nest code. How much can I fiddle with that without losing the point of working with Brain Modeling Toolkit? Either of you can take that one.
Yeah, so I think you should be able to do that. We've actually done this before, something similar before, not with synapses, but with custom cell types. For instance, at the Allen Institute, we have our own models, which are called glyph models. We have version of these glyph models available in Nest, and it's fairly easy to actually run these models, these glyph models, using BMTK. So Anton mentioned the V1 point neuron model that was actually run using custom Nest neuron types. I'm not sure how synapses would work, but I think it should be able to work the same.
Yeah, and just to add to that, maybe you can give it a shot, and if you have any questions, let us know, and we'll be happy to help.
Great. I have a question from YouTube. This one was during Kale's portion of the presentation. Can you elaborate on the dynamics parameters?
A little bit. So you can pretty much create whatever types of, SONATA sort of allows you to create custom cell dynamics parameters, however you fit. One thing SONATA does is it actually tries to accommodate for multiple simulators, not just neuron and Nest. One thing we can do is you can go to the Allen Cell Types database. For individual cells, you can actually download dynamic parameters as JSON files and just plug them into BMTK, and BMTK will automatically recognize them. So if you want to create a model from the Allen Institute. Another thing, it also, as Anton said, we actually have support for NeuroMel. So if you have like a cell with all its ionic channels in NeuroMel format, you can, BMTK will be able to recognize it and run models like that.
Great. This is another one that came in during Kale's portion. Do you have the possibility to randomize neuronal and synaptic parameters over neural populations, given an a priori set of probabilities?
I don't think we've actually tried that. You can randomize it during the building process. So one thing we sort of built BMTK and even SONATA based on is building these very large, we want to build and share these very large network models. So during the building process, you can randomize it, but then it's pretty much going to save parameters statically. There might be some ways to sort of do some more randomization, but I would say it's not, if you can think, I would say like contact us and we'll think about it, but maybe sort of.
We have a technical question on Windows. Is Neuron only available for Python 2.7?
No, I use Linux, and I've never actually used Neuron on Windows. And it should be available for Python 3.6 and above. It is available, I know that.
Great. Now, Anton has been answering a lot of questions by typing those. So thank you so much, Anton. Do you have any particular answers that you want to elaborate on briefly or highlight?
Well, I think a lot of questions were very specific, but yeah, we really thank you for the interest. There was some questions about documentation. We have a fair amount of documentation and some tutorials too in the links that we've shared. It may not be, completely comprehensive yet, but we are certainly striving to do as much as we can. If people have questions about any specific functions or functionalities, please let us know. email us, create tickets on GitHub, and we'll be very happy to work with you.
And please visit us on our community forum as well at community.brain-map.org, where we'll also post all of the answers that we've covered today. And then we have a couple of questions that we are out of time and we're not going to get to, but we will get those answers to you there. So thank you, everyone. We are out of time. Thank you for joining us. Again, the video of this will be available on YouTube to watch later. And I hope you'll join us for some of our upcoming tutorials. We have one at the end of February, and we have a symposium and a series of additional tutorials in March. The information on how to register for those is available on the Allen Institute website at alleninstitute.org, and then click on events. Thank you for joining us today, and we will see you at a future event.